Hello my dear friends, welcome to my YouTube channel, Biology with Ritu. I am Ritu Rawat and I hope you all are doing great. So today's video is gonna be about how to make a monohybrid cross and dihybrid cross. Before I start this video, I would like to say if you guys haven't subscribed my channel, so please subscribe to my channel and don't forget to like the video. And yeah, make sure to press the bell icon to get the notified. So finally, let's get started. So first, let's have a quick intro about Gregor Mendel. So basically, Gregor Mendel was a famous biologist and he is also known as the father of genetics. He conducted his experiment on garden green pea plant, that is Pisum sativum. And he conducted his experiment for 7 years before proposing the law of dominance. We will study about his 3 laws in the next video. Now the question rises here that why he chose pea plant only and not the other plant. So here are the few reasons. First, easily available. Second, short lifespan. Third, large number of progenies were produced. Fourth, cheap. And fifth, 7 distinguishable characters. So let's have a look on the 7 characters. Now let's move on to the main topic of today's video, that is monohybrid cross. So this cross is made between 2 individuals of same species considering the transmission of one character only. Let's take an example of plant height. So first, we should know that plant height is the character and tall and dwarf both are the traits. First, we write the character on the left side and make sure that all the headings should be written on left side, like in this sequential manner. Then we write the traits as the parents, like this. Then we write the gametes. So gamete is always written inside a circle, and we already know that due to meiosis during gametogenesis, the 50% gamete is transferred from the father and 50% from the mother. Now we write the F1 generation and the F1 generation. Here F1 indicates flyel. We call it flyel. As we can see here that the progeny in F1 generation is tall. This is so because the pure TT means capital TT is dominant over small tt means dwarf. Alright, it means that the progeny produced in F1 generation will be tall. And now we do the self-crossing like this. Again, we write the gamete. We write the F2 generation, and for this first we have to draw a box. Before we go ahead, you should know that this sign is used for male and this sign for female. So here, as we can see that there are 3% tall progenies and 1% dwarf progenies. These are in ratio. Now we write the phenotypic ratio, that is 3:1. You should know that phenotypic ratio means the external feature that can be seen morphologically. So here 3:1 indicates 3 for tall and 1 for dwarf, means tall ratio dwarf. Now we write genotypic ratio, and that is 1:2:1. And genotypic ratio means the genes coding for the phenotype. So here 1:2:1 indicates means 1 for pure allele, tall, means capital TT, and 2 for hybrid tall means 1 capital T and 1 small t, and 1 for pure allele dwarf means small tt. Finally, we are done with the monohybrid cross. So now let's move on to the next topic, that is dihybrid cross. So basically it's a cross made between two individuals of same species like before, but considering the transmission of two different characters. So this is similar to the monohybrid cross to some extent. So let's have an example of seed color and seed shape. So let's write the character first. Now similarly write the parents. I hope you guys already know that for the recessive traits we write in small letter. Now we write the gametes. And make sure to make a circle whenever we show the gametes, because when we present it inside a circle then it makes a correct sense. Now we write the F1 generation. So here in F1 generation we get the round and yellow progeny, alright. Now again we will do the self crossing like this. Now we write the gametes. So here for the gametes, some students get confused while writing the gametes as it's little bit typical than the monohybrid cross, so watch it carefully. I know it seems somehow messy, but you guys don't need to worry about it. I'm here to help you because I'm gonna tell you an easy trick. So what you can do is write the capital R with capital Y, then write capital R with small y, then similarly small r with capital Y and small r with small y, like this. And finally when you are done with this, then write down in the other side as well, like this. I hope now you won't get confused after this. Now write after generation. So this is little bit typical, but I'll try my best to make it easy for you. So let's start making a box first and write down in sequences, alright. So make a box first, and this sign is used for male and this is used for female. Guys, try to write down carefully so that it won't look messy. Make sure to draw a neat and clean box, alright. Now finally we have came to the last 2 points, that is phenotypic ratio and genotypic ratio. So for phenotypic ratio we write 9 ratio, 3 ratio, 3 ratio, 1, alright. And as I've told you earlier that Phenotypic ratio is the morphological feature, so means 9 indicates round and yellow seeds and 3 indicates for round and green seeds and 3 indicates for wrinkled and yellow seeds and 1 indicates for wrinkled and green seeds. So these are the ratio and you can count here. And finally, we write the genotypic ratio. And genotypic ratio is 1:2:1 1:2:4:2:1:2:1. It's a little bit longer, but you don't much about this ratio, just learn it. And the simple trick is, first write down the genotypic ratio of monohybrid cross, that is 1:2:1. Then make it double, like 2:4:2, and then again make it half, 1:2:1. Alright, and finally you are done with the dihybrid cross too. And yeah, one more thing I would like to add here is that if you guys want my handwritten notes of this chapter, means chapter number 7, then you can write down in the comment section. And if I get more than 10 requests, then I'll add the PDF of this chapter in the description box. So guys, now I hope you have understood both the crosses, as I tried my best to make it easy for you. Yeah, now let's end today's video. I'll catch you soon in the next video. Till then, bye bye. Have a good day. Take care. Thanks for watching this video. Have a good day.